Hi guys, so now that we are completely done with transcription and translation, we've talked a little bit about some of the PCR technology, we're going to backtrack a little bit and look at another division of genetics. So you may remember this figure from chapter 1 that described the relationship and the difference between transmission genetics, molecular genetics, and population genetics. So we've spent the first half of the semester really investigating molecular genetics. So that had to do with looking at the chromosomal variation that exists in different species, taking a closer look at the chemical nature of DNA like we did in chapter 10, discussing how DNA is actually replicated, synthesized, and copied in chapter 12, and then more recently how that DNA material is copied into messenger RNA and then further translated into amino acid sequence. So all of those 5 different subjects really have to do with molecular genetics, which is studying the way that genes are actually expressed in an organism. Where we're headed now is looking more so at transmission genetics, and usually genetics classes start with this, but it's been our experience based on last semester that the transmission genetics really need a little further illustration. So we're hoping that now you guys have a good handle on the molecular genetics, that looking at transmission genetics, you'll have a little bit better foundation. So we're going to start with chapter 3, which is where we're at today, and we're going to look specifically at how genes are passed from generation to generation. Then we'll expand those principles in chapter 4 and talk more about how sex is determined and some of the diseases and characteristics that are related to the sex chromosomes. And then finally in chapter 5, we'll talk about some of the modifications and the extensions that happen of those genetic principles that we're going to talk about now in chapter 3. So before you guys begin chapter 3, you need to make sure that you have a really, really good handle on these genetic terms. So what is a gene? How does an allele differ from a gene? What is a locus? What is a genotype versus a phenotype? A heterozygote versus a homozygote? And then how do we measure these different types of characteristics? So like if we evaluate it as a phenotype or a characteristic, etc. So just to make sure that we're all on the same page as we begin the discussion about chapter 3, you guys all know what a gene is. It may also be referred to as a loci, but what we need to make sure that you guys have a good handle on is what an allele is, and it is an alternate form of a gene. And this can occur either in one or two different fashions. And we've got an example of that over here in this figure to the right, where we have one chromosome from mom, one chromosome from dad. And the chromosome from mom has a big R allele for this gene location. And the chromosome from dad has a little R allele for this gene location. So there's still these are both the same chromosome, and there's the same gene that's here, but one may code for blue eyes, one may code for brown eyes, just depending on what traits were passed on from mom and dad. So that's going to become really important as we start talking about inheritance and creating variation within genomic species. So the father of genetics was Mendel, and as you guys already studied a little bit in chapter 1, and he really was responsible for creating this avenue called transmission genetics. We also hear it called as Mendelian genetics. And what Mendel did was that he used scientific theory to test his hypotheses. So he didn't just theorize about how things existed like Aristotle did, that when we looked at chapter 1, but he actually carried out full experiments. So the experiments that he carried out were on peas, and he specifically studied a couple of different traits. He studied the color of the seeds, the shape of the seeds, whether they were round or wrinkled, the coat color of the seeds, whether it was a gray coat color or a white coat color, the pod color, the shape of the pod, and then the flower, both the position and the stem length as well. So he was looking at 7 different phenotypes or 7 different traits and was really making conclusions about how the genetics determined these different phenotypes. So, some of the take home messages from what Mendel found was that there are 2 alleles that are going to be contained in the system, and when they separate, um, is when the gametes are formed, and one allele is going to migrate into each gamete. After that gamete has been designated, then fertilization is going to occur, and 2 alleles are then going to come together and create the genotype, which is eventually going to lead to the phenotype through transcription and translation. He also determined that the dominant and recessive phenotypes during the first part of his work, where he identified that one allele ended up being dominant over the other allele, which was termed recessive. So Mendel's first law is really what we're going to talk about in chapter 3, and it's the principle of segregation. And this law is really looking at how alleles separate or segregate in a way where each gamete is equally likely to contain either member of the pair of heredity determinants. So, in other words, that is going to really focus on how a chromosome is going to replicate, and once it replicates it has a complementary pair, and then as we go through the meiotic process during prophase 1, we're looking at how those different chromosomes segregate out. So and we're also going to be talking about how we create variation in some of those different chromosomes. So this leads back to a little bit of the discussion that we talked about in Chapter 8, where crossing over can take place, as well as exchanging genetic material. So the chromosomal theory of inheritance that Mendel was also responsible for was stated that genes that are located on chromosomes and they are segregated into cells during meiosis. So Mendel was, his first work really looked at determining the genetic reasons for single loci crosses. So single loci is really referring to single genes that are coding for a single trait. So the trait that we're talking about here is height in our pea plants. So you can either have a tall plant or a short plant. And Mendel was really one of the first ones to actually design this, what we call today these Punnett squares, where you have one genotype up here at the top of the square, one genotype over to the left of the square, and then you just pair up the different genes to get the phenotype. So if we have a big T and a little t, we bring those together. Our genotype then becomes big T, little t, and we know that big T is dominant over little t, so you're going to get a tall plant. Same scenario for this square as well, but when we get down to the bottom squares here, we're going to get inheritance of a small t from mom and a small t from dad, and that's going to result in a short plant. And so the two phenotypes are tall and short, and the phenotype is height, but the two results of those phenotypes based on the allele combinations, big T or little t, can result in either a tall or a short plant. So in order to find out what a genotype is going to be, we use the rule of probability. And probability is going to be a tool that helps us determine the outcome of a genetic cross. So you guys are going to be doing quite a bit of calculations and math in this chapter. So there's 2 major rules that are associated with probability. The first one is the multiplication rule, which I'm sure you guys are all familiar with at some point. And this is going to calculate the probability of 2 or more independent events occurring together. And it's going to be calculated by multiplying the independent probabilities. So when you're reading word problems, you know that you need to use the multiplication rule if a problem specifically uses the word and. And the multiplication rule can also play into a more complex probability calculation, which we call binomial expansion. And we'll talk more about that when we get into the nuts and bolts of the chapter. The second rule is the addition rule, and this is going to assess the probability of any two or more mutually exclusive events. So an event that can occur exclusive of a second event. And this is going to be calculated by adding the probabilities of the two events together. So the key phrases that you look for in these problems are or and either. So to visualize these two rules a little bit better, the multiplication rule is going to multiply the probability of the events happening independently of one another, whereas the addition rule is going to calculate the probability of two events that are going to be mutually exclusive. And mutually exclusive, again, means that one event is going to exclude the other probability of occurring. So in the last couple of slides, we've looked pretty in depth, or not in depth, but very briefly at single loci crosses. But Mendel also did some advanced work looking at multiple loci crosses where he was assessing two traits. So, for example, here in this scenario that is in your book in chapter 3, Mendel was really assessing the color of the seed as well as the shape of the seed. So the color was either yellow or green, and the shape was either smooth or wrinkled. So two phenotypes, two traits, two alleles within each gene, and then there's two genes that code for both of those traits. So R is going to refer to the color, whereas or the first position is going to refer to the color, and the second position is really going to refer to the shape. All right, some other tools that we're going to be using to estimate probability of certain events happening is a chi-square test. And for those of you guys that have been through AGRI 490, this should not be a foreign concept to you. But the goodness of fit chi-square test is where we're going to assess the results that we observed versus what we expected in a population. So we're going to be trying to assess the probability of a certain phenotype occurring. So in this case here, we've got a purple flower and a white flower. We want to know if we cross them, what is the probability that we're going to get a greater percentage of purple flowers over white flowers. So to do that, we're going to go through a series of mathematical equations, and don't worry, we're going to go through this very in-depthly, and then we're going to assess our answer that we get from these multiplication or these equations to a chi distribution chart, which you can find in your book as well. And based on that, you're going to make an assessment as to whether the chi-square test was significant or not. All right, so that is the end of the prep quiz for the prep lecture for chapter 3. Don't forget to take your quiz, and we will see you guys on the 31st.